Hello viewers, my name is Dan Larry. In this class, we'll be looking at the topic nutrition. Nutrition. What is the meaning of nutrition? The first thing that will come to your mind when you hear nutrition is food. And apart from food, we also look at feeding. That means taking in food, or we call it nutrition. So what is the meaning of nutrition or feeding? This is the supply of vital materials or nutrients to the cells of an organism for growth, respiration, reproduction, and other activities that pertains to life. Meaning, when you see a living organism, one of the things that makes the living organism to survive is food. So the intake of it makes that living organism grow, makes it to breathe, take in oxygen, breathe out carbon dioxide, makes it to give birth. So organism can give birth because it takes food that will give it life and other activities. Food is the source of nutrients. If you say you have nutrients, this is coming from food. Food supplies energy to the cells, and that's why cell can carry out different metabolic activities. That's anabolism and catabolism. That means building up and breaking down. Nutrition is one of the characteristics of living things from Mr. Niger D. So where N is nutrition. So this is the process by which organisms obtain, they get and utilize. That means they get the food and they make use of the food. Utilize materials from external environment, meaning that food you get it from the environment and you use it for your metabolic activities. That means the breaking down and the building up. Just like a house, you get sand, use it to build the house, and you break down some things in the process. So this metabolic activities means respiration, growth, excretion, and reproduction. When you hear metabolism, that's all the chemical processes associated with living organism. So like I said, it's divided into catabolism and then anabolism. So when you hear catabolism, it has to do with how complex substances will be reduced or decomposed into simple substances, and at the same time energy will be released, so which will be used for the organism. It will be available for them to do anything. On the other hand, anabolism or anabolic process, this one has to do with building up of complex substances, so with the absorption of energy. That means the energy that was released during catabolism will be absorbed here. The building up that took place, or the breaking down that took place in catabolism, will now be built up in anabolism. Energy that was released in catabolism will be absorbed in anabolism. So metabolic reactions, they are controlled by enzymes. That means for building up and breaking down to take place, enzyme must be involved. The types of nutrition we have: auto and hetero. Autotrophic. Auto means independent. Hetero, getting from different sources. Meaning that if you look at plants, they are independent when it comes to food. They wait for food to meet them. That's for sun, rain, and lights to come and meet them there. So it means they are autotroph. That trophy has to do with feeding, so independent feeding. But hetero, from different sources they get their food, like animals going to different places. Let's look at autotrophic nutrition. Like I said, the plant majorly. So it's a type of feeding in which organisms will manufacture their own food. They don't look for food from anywhere. They are independent. So different types of autotrophic, like photosynthesis, it means they will depend on photo. That photo is light, sunlight. Chemo, that's chemicals. So it means through chemicals they make their food. So for the photosynthesis, that's they will need carbon dioxide from the air, water from the land or where rain is falling, and sun will be shining for photosynthesis. They will use their chlorophyll to absorb sunlight. That's those green plants. And at the end, they will form their glucose, and oxygen will be released as a byproduct. So that's photosynthesis. But chemo, from chemosynthesis, chemical, they will manufacture their own food from simple inorganic substances. Inorganic means minerals, maybe carbon dioxide, water, hydrogen sulfide. That's the inorganic. something that is not from plant and animal, they will get it. After getting that, they will use the energy that will be released in the process during the chemical reaction, so they will use it to form their own food, and that's chemosynthesis. So all these are from inorganic substances, so which are chemicals, so used to produce energy. So there are different bacteria. We call this one sulfur bacteria. So they can manufacture their own food by oxidizing hydrogen sulfide. That means they will remove the hydrogen and they will get their sulfur. So, and that's why we call them sulfur bacteria. So the chemical energy that is capable of bringing about the combination of hydrogen sulfide and carbon dioxide in a soil to form sugar, water, and sulfur. So that one too is chemosynthesis. So meaning that we can still get the same glucose gotten by photosynthesis. We can also get by what? Chemosynthesis. That's through the chemical hydrogen sulfide and carbon dioxide. So in the soil, plants, or let me say organism, plants is also involved since it's still under autotrophic. So plants can make use of the chemical hydrogen sulfide and still absorb carbon dioxide, and at the end produce their glucose. So that's, and then from the glucose, so which is the simple sugar, they can still get other substances like protein, oil, vitamins, and so on. So there are still another example of chemosynthesis apart from hydrogen sulfide, which is through sulfur bacteria. We also have one of nitrogen bacteria, which we call nitrosomonas. So from ammonia with oxygen, they can get this HNO2, and the HNO2 can in turn become HNO3. So all of these are from chemicals. So they will get their energy and then use it to combine with water, carbon dioxide, and finally they will still get their sugar, that's the glucose. Now let's move to heterotrophic. Remember, hetero means different, meaning that from different sources you can get food, and that's majorly those organisms that always depend. They are not independent, they depend. Like fungi depend on food from different sources, bacteria, especially those organisms, those food that for organisms that are dead. So they can get their food from anything that is dead. Parasitic, they will enter into organism and then begin to benefit from them, like parasitic flowering plants. So they are heterotrophs. Now, looking at heterotrophic nutrition, we have holozoic, saprophytic, symbiotic, parasitic. When you hear holozoic, the zoic is from zoo, animal. So meaning that holozoic has to do with how animals will take their food. So they will eat it, they will digest it, they will absorb it into their body. So they obtain it as solid organic. So they take it in form of organic materials. So they will digest their food, as I said. So an example of holozoic, those ones that are getting food from other sources. That means they are dependent. Like herbivores, they are holozoic animals that depend on others. So, and from the herbi, mean that from plants they get their food, like sheep. Carnivores, anything carnivore, that's they eat flesh. So they depend on flesh of other organisms like leopards, dogs. So they are carnivores eating prey. They capture their prey. So they are called predators. So while those feed, so those things, those that feed only on dead animals, they call them scavengers. So it means they are eating on flesh. So either flesh that is alive, so from carnivores, predators, or those ones that are dead, that are scavengers. Omnivores, omni is both, or whole, meaning that this one can feed on both plants and animals like man. They are omnivores, so they are still under holozoic. Now we move to saprophytic. In saprophytic, they depend on anything that is dead or decaying organic material, but enzymes must be secreted if they will get their nutrient from the dead substance. So enzymes must be involved, and their food digestion will take place outside. That means they would have digested their food outside from that dead organism before it gets to their body, and that's what we call extracellular digestion. So those organisms doing that, we call them saprophytes, majorly the fungi like mucor, mushroom, yeast, bacteria. Now we move to symbiotic. In symbiotic nutrition, it means both will benefit. They benefit from each other. This organism will benefit from this. This one will also benefit from the other, like algae and fungi, which form leaching. So the fungi, so they will provide moisture, that's water, provide mineral. They will provide support to what? Algae. But algae, they will manufacture food by photosynthesis for the fungus, so and itself. So another example is found in the hermit crab and sea anemone. Both of them they benefit from each other. Herbivorous animals and cellulose. So that's digesting bacteria that will live in the cecum and what colon of herbivores. Now that leads us to parasitic nutrition. When you hear parasitic, that's parasites. So one will be hosts. So it means the host is the one that a parasite will enter his body and be benefiting from that organism. So that process is parasitism. So if the body of the organism is outside, so where the parasite is staying, if it's outside, that's ectoparasite, but if it's inside, endo. So for ecto, those ones that are outside, like lice, you see lice on the body of organism, that's the air. Tick in the blood, sucking blood of dog, bed bug, so they are outside. Flea, mites, but endo they are inside, like the plasmodium of mosquito, hookworm, ascaris, all those worms, tapeworm, those ones that disturb the intestine, liver fluke. So they are all endoparasites, staying inside to take whatever they need. In short, they stay on the small intestine of their host and then get the digested food as it's coming. So this is tapeworm and endoparasites. So having what we call rostellum, having hook. So this is the rostellum, the mouth here is the rostellum, the curved part here is the hook. So it means they use it to stay on the body of the intestine. And these are the suckers. The one that look like eyes are the suckers. Then this the mixture proglottids. So and then the right proglottids. So the one that will form another tapeworm. So that's that on that. Then apart from animal parasites which have been mentioned, there also exist some parasitic plants. Plants can also be parasite, they can also stay on the body of their host. An example of some parasitic plants, we have Doda. Doda is Casita filiformis. We have Misuto. So all these they hang on the body of organisms, so on the body of plants for their survival. So they will obtain their nourishment by developing special organs which we call ostoria, that's suckers, and are sent to the host vascular bundle. They will go and suck it out. So you can see their structures there. Adaptation of parasites include, that means how do these parasites survive in the body of organism? So they have organs of attachment, what they will use to attach, like hook and sucker in tapeworm, they will use it to attach. So and osteria also, they use ostoria, that's Misuto. Misuto will use ostoria to hang on the body of the organism. Some they have simple body structure, so since they do not have to search for food, so they don't have big structure, so they can easily enter. Resistance to digestion, so that means when they are in the body of in the intestine, like intestinal worms, so their body will not be digested with the food that is being digested in other organisms, so they will rather just take the digested food into their bodies, like roundworms. Now the next is production of large numbers of eggs, so that's what makes them survive. So their eggs will keep reproducing, producing new young so that they can survive, and the infection of new hosts, so like tapeworm. So the existence of an intermediate host in some cases, so like malaria parasite, they have intermediate hosts, so they can host many things, like also guinea worm. Now we also have what we call carnivorous mode of nutrition in plants. When you hear carnivorous plants, so these ones they have green leaves and are able to manufacture their own food. They carry out photosynthesis. So they have roots. Using the root to absorb water and mineral salt from the soil, they usually grow in places with little nitrogenous salt, and they will use insects or other small animals as source of nitrogen. So that's why we call them carnivorous plants. So once they take the insects, they will get nitrogen from it. So they have device that is modified or modified leaves, so with high color, scents, sugary liquid, sticky wet surface for attracting and trapping insects. So once insects see those plants with all these colorful things, they come immediately, they trap them. So once the insect is trapped, the plant will secrete enzyme, and that will be used to kill and digest that particular insect. So soluble nitrogenous compounds they get from their insects, so they will absorb, assimilate it, so the plants will enjoy it. So carnivorous plants they are called insectivorous plants because they are eating insects. An example, we have butterwort, which we call pinguicula. We also have bladderwort, which we call utricularia. We have sundew, which is drosera. We have Venus flytrap, that's Dionia, pitcher plants, Nepenthes and Sarracenia. So all these are carnivorous plants eating insects. So you can see the flower. So they are using it to attract insects. So this air is very sensitive. So sensitive, once there is insects, so they should be ready to trap that insect. So this is a pitcher plant also with its leaves. So using this leaves to cover any insects that enters them. So there are some plants that grow upon the surface of another plant without causing harm to such plants. So these plants we call them epiphytes. So they will just stay on top of another plant, but they will not affect them. Like Moses and orchid, they are epiphyte. Moses gently, just like Moses on water in the Bible. So this was also Moses can stay on the body of another plant. The same thing orchid. Orchid will just be there on the other plant without affecting it. So they will not grow. So they do not grow osteria, and therefore they are not parasitic. So that's that on that aspect. So another thing we consider today is let me scroll down so that you can see the next. So plant nutrition. How do they eat? That's by photosynthesis. We have said that earlier. So through especially those ones that have green plants, they will take their carbon dioxide and water in the presence of light, and they have their chlorophyll in their chloroplast, use it to absorb the glucose or form glucose and produce oxygen as their byproduct. So which is a simple sugar. The glucose is a simple sugar they produce. Although all the green parts areas that contain chlorophyll of a plant can carry out photosynthesis. So the leaves are the primarily photosynthetic organ. What will do the major work? So the major site of leaf where photosynthesis occur, so they call it palisade mesophyll. So it's at the palisade mesophyll of the leaf. That's where they will absorb the sunlight. Why? Because they contains more chloroplasts. So palisade, unlike the sponge, so they have more chlorophyll than the spongy layer, other part of the leaf. The palisade cells, they are also located just beneath the upper epidermis. So it means they are outside, very close to the external part of the leaf. So where the chlorophyll could absorb more sunlight. So take note that apart from chlorophyll, there are some other pigments which are capable of absorbing sunlight energy. Example includes we have carotenoids. Carotenoids. When you hear carotenoid from the word carotene. So carotene. So carotenoid, like they are orange in color. Xanthophylls, these ones are yellow in color. You can use X and Y. X xantho, Y yellow. So these ones are yellow in color. Carotene from carrots, like you can just say orange in color, even though carrot is not orange in color, but just carrot and orange. So you can say orange from raw carrot, orange. So orange in color. And then anthocyanin. This one is bluish, like cyano. You know, cyano has to do with blue green. So they are bluish green. So these are accessory pigments and are masked by chlorophyll green, which is the main photosynthetic pigment. So it means all these colorful things also, they are always attracting. So they're just like green, we attract the sunlight. All those ones also attract, just that the green one is dominating others. So it's always visible when leaves fall and die. So here is transverse section of the leaf showing chloroplast containing chlorophyll. So these are layers of the leaves where we have the epidermis, the palisade chloroplast, the xylem phloem under the vascular bundle, the S-space under the spongy mesophyll, then the lower epidermis where we have the stoma, the guard cell. So this one has been explained in our previous topic. Okay, so another thing we need to understand here is adaptations. How do leaf adapt? How do they adapt for photosynthesis? One, they have broadleaf lamella. So the broadleaf lamella means they are very wide. So, and that will give them enough surface, large surface area to absorb enough sunlight. They also have what we call stomata. This one will allow carbon dioxide to diffuse into the leaf and oxygen to diffuse out. That's why they are able to survive. They also have what we call epidermis. Epidermis is thin and is transparent. It can allow more light to reach the palisade cell where there will be photosynthesis. So that's epidermis because it's thin. The palisade cell, it contains many chloroplasts. There are a lot of chloroplasts in that which will absorb light. There's a thin lamella This one will provide just short distance for carbon dioxide to diffuse into the leaf. There's a thin corticle that is made of wax. So this one is just to protect the leaf from infection and prevent water loss without blocking out light. That means despite the fact that it's a covering, yet it doesn't stop light. We also have what we call xylem. This one is specialized tissue. It transports water into the leaves for photosynthesis. There's also phloem. This one will transport manufactured food to other parts of the plant. We have what we call network of veins. This will support the leaves and transport water, mineral ions, and sucrose. That's the sugar. Then we also have chlorophyll. The chlorophyll, this one will absorb sunlight energy and transfer it into what? Chemical energy. So the process of photosynthesis will divide it into light and dark reaction. So when we say photosynthesis, it occurs in the day. So because there is light, so that's we have light reaction. This takes place only in the light. And this light reaction is divided into 4 steps. So the 4 steps of light reaction: number 1, chlorophyll is energized. Remember that's where it must start from. Chlorophyll that will make sure it traps sunlight, so it's energized. So the chlorophyll will absorb sunlight energy and then it becomes energized. So once it receives, remember energy is from the sun, so chlorophyll will absorb sunlight for energy. Now once the sun comes, there's what we call photolysis of water. Any water that is in the plant, so the sun will now split it. That's what we call photolysis. So the energy absorbed by the chlorophyll is used to split water molecule into hydrogen ions and hydroxyl. You know water, H2O, so it means you can get H and you can get OH, so to just split it. That's photolysis. So the OH you see there, which we call hydroxyl ion, will give up its own negative electron to the chlorophyll molecules, and then it will form water and oxygen. So that means, when you combine OH plus OH, so it will give you H2O. So this is it here, and oxygen. So combination of OH with another OH, so it will form water and oxygen, meaning that H, the second one is there, and OH2O. While this O will go and form oxygen. So meaning that after photolysis of water into hydrogen and hydroxyl, combination of hydrogen will give you water and then oxygen. Now what do we need the oxygen for? So the oxygen is the byproduct, and that's why we say plants will breathe out oxygen. Now step 3, the hydrogen that was the other product. Remember, hydroxyl turned to water and oxygen, but what of the hydrogen? The hydrogen is transferred by NADP. When you hear NADP, that's nicotinamide adenine dinucleotide phosphate, NADP. The way we have NACP, so triphosphate. The way we have tri, we also have, okay, sorry, that one is for ATP and ADP, but here now we are looking at NADP. Anytime you just see NADP, first thing that should come to mind is hydrogen. So because hydrogen works with NADP, so hydrogen is transferred by NADP. So the hydrogen atom released during photolysis of water, like I said, is picked up by this coenzyme. Coenzyme means a supporting enzyme. It always supports enzyme in breakdown process. So meaning that the hydrogen, so will be picked up by this coenzyme. So what will they use it to do? This is to prevent the escape of hydrogen. We just trap that hydrogen. That means this coenzyme will trap the hydrogen atom from the cell. So, or it should recombine with what? Oxygen. It should recombine with oxygen to form water. So they will just make sure the hydrogen is not wasted. So that's why the NADP, which is the coenzyme, will pick up the hydrogen. So and then it can keep it, or it should give it to oxygen so that it can form water that is still needed. This same NADP again, so when reduced, that means addition of hydrogen is reduction. So once it has take the hydrogen to itself, it will form NADPH2. So that means 2 molecules of, or 1 molecule of hydrogen is H2. So it will form NADPH2 and it will now become electron carrier. It will serve as electron carrier of hydrogen ions. So meaning that it has taken the second hydrogen. Now the last step, formation of ATP. That's adenosine triphosphate. So the chloroplast contains ADP. That's adenosine diphosphate. That's what we have inside chloroplast. Chloroplast of plant, they have ADP. So what will it now do now? So there will now be an energy, we call ADP, adenosine diphosphate, which is an energy carrier. Energy carrier is inside the chloroplast. So the extra energy that is not used in the splitting of water. We said energy from sunlight will split water. So this means there is still another energy. So that is remaining, that is not used in splitting of water molecules. So it will be released to what? ADP. So the sun will just say, okay, the remaining energy, let me give it to ADP, so that ADP can form ATP, so which is used in the dark reaction. So that energy, since there will be no energy at night, so the energy in the day that is remaining, so will be given to ADP, so that you turn it to ATP, and that's what will be used at night for the dark reaction. Now the light reactions take place in the grana, that's granium singular form of the chloroplast. Now what are the major products of light dependent stage of photosynthesis and the importance of the products? So the product during the light process is what? ATP. Remember that I said the remaining energy from the sun will be given to ADP to form ATP. So that's the products here. And what is the importance of this ATP? That's all, is to generate energy for the dark stage. So during the dark stage it will use it. Then what of the hydrogen that is remaining from the water that has been splitted? So that hydrogen is another product. So just that this hydrogen will later be picked up. So the hydrogen will be picked up by NADP to turn it to NADPH2. And since it has formed NADPH2, there will also be H plus. Remember NADP. So the NADP, so as soon as the hydrogen comes, so it will stay beside it, but it will be double. So just like in glycolysis, there will still be an hydrogen ion, which is a byproduct. And what will this hydrogen ion be done? What will it be used for? So it will combine with NADP to form NADPH2. Like I said earlier, that hydrogen from the splitting of water to form NADPH. So the hydrogen after combine with NADP to form NADPH2. Now the hydroxyl ion. Remember we said splitting of water will have hydroxyl ion. So this one will form water. Like I said, it will recombine. Hydroxyl and hydroxyl will form water and oxygen gas, which are released as byproducts. The NADPH2, after hydrogen has come together with NADP, so that NADPH2, it will reduce, or is a reduced nicotinamide adenine dinucleotide phosphate. That means the NADP has been reduced by the hydrogen. So that's why we call it a reduced. Once you see that H has been added, it means it is a reduced form of nicotinamide adenosine diphosphate dinucleotide phosphate. So what is it used for? This is to generate hydrogen ion. So immediately hydrogen enters here, there will be what? Hydrogen ion. So it has generated hydrogen ion, which is to reduce carbon dioxide. So once hydrogen ion has been generated, it will reduce carbon dioxide to form carbohydrates. So to form carbohydrates in the dark stage. So meaning that in the dark stage, the energy that is needed will be coming from the light stage. So the remaining energy that is formed, so it will reserve it, will conserve it from the ADP to ATP. That's one. Then the hydrogen also, we said the hydrogen ion to generate hydrogen, which is to reduce carbon dioxide to form carbohydrates. There will be no enough of all these things at night, so that's why they are reducing it, they are storing it. So to reduce carbon dioxide and turn it to carbohydrates. So it's that carbohydrate that will now be used in the dark stage. Finally, oxygen gas, which is this other byproduct. So after we have formed water and oxygen, so that oxygen now will be given up in the atmosphere for animals or other organisms to use for respiration. Now we move to dark reaction, which we call Calvin cycle. This reaction does not require light energy, so that one has been done earlier. So carbon dioxide is reduced by combining with two atoms of hydrogen provided by a coenzyme. Remember that the coenzyme NADP has already taken hydrogen in the light reaction, so now we have NADPH2. So it means the hydrogen that is now stored here now will now be used to reduce carbon dioxide. So the carbon dioxide, so it will be reduced. So from the hydrogen that was stored, in this NADP, and this will now form sugar. So that's after undergoing a series of stepwise reaction. Each step is controlled by a specific enzyme. So the energy needed for formation of sugar is provided by ATP. Remember that ATP has been formed in the light phase, so that energy that is needed, so it will be used for this dark phase. So from the light reaction, so the dark reaction takes place in the stroma of the chloroplast, So this is a carbon dioxide that is needed at night, hydrogen that was gotten in the day, so we come together with it, reducing it to the glucose we need and water. So meaning that that water also can come back to add with another carbon dioxide to get more glucose, and oxygen will be released. Now, what are the materials and conditions necessary for photosynthesis? If photosynthesis will take place, we need water, we need carbon dioxide, we need light, and the temperature must be suitable, and there must be chlorophyll, that's green plant. What are now the factors that can affect rate of photosynthesis, the way it will take place? External factors, the intensity of the light. Is there enough sunlight? Is there carbon dioxide? There is enough concentration? Temperature, is it high or low? Pollution, hope the environment is not polluted, and water. These are the factors that will affect photosynthesis. Then for the internal factors, we have hormones, chlorophyll or presence of chlorophyll, chloroplast, enzyme, number or state of stomata. All these are within that will take place, that will allow, that make us to know if photosynthesis will take place or not. So the hormones has a way of making sure photosynthesis take place. So we have different hormones, like we have auxin, gibberellins, and so on. So they will help us to know if there will be photosynthesis. Then chlorophyll, so that's for greenness, to absorb sunlight. Chlorophyll, so then enzyme, so that will make sure those things that are some enzymes, so they will make sure certain things are broken. So they will make help in the breaking and also in the keeping of some substances. The stomata, opening the way for carbon dioxide and so on. Now, what's the evidence of photosynthesis? The first main product of photosynthesis is glucose. That's how we know that photosynthesis has taken place. Once you can see glucose, so it is immediately converted into starch for storage. So that glucose formed, it returns to starch. That's how to know that photosynthesis has taken place. Now, presence of starch in the leaves will show that photosynthesis has occurred. Some plants, especially monocotyledons, do not change their own sugar into starch. That's monocot. They don't change their sugar to starch. If such leaves are tested for starch, a negative result will be obtained. So if you say you want to do test for starch, don't go and take maize leaf, because the glucose in them wouldn't have been converted to starch. So in such plant, part of the food synthesized are converted to soluble forms and carried in the phloem vessels to storage organs. So what's the fate of food manufactured by green plants? Glucose is the first stable product of photosynthesis. Manufactured food like glucose will be translocated to various parts of the plant through the phloem. So this can be combined with other elements such as nitrogen, sulfur to form proteins for growth. So it can be converted to starch and stored. So it is used in respiration, metabolic activities, or broken down into carbon dioxide and water to produce energy. So other products, we have lipids, we have cellulose. So this also can be manufactured from glucose. Once you get glucose, you can get some other compounds. So is photosynthesis important? Yes. Green plants, they manufacture their food through this photosynthesis. So if there's no photosynthesis, it means green plants cannot get their food. All other organisms, they depend on this process also, as their ultimate source of energy is this same plants. So they feed on plants. So meaning that if there's no photosynthesis, there is no food for herbivores. Carbon atoms present in the organic molecules of organisms, they derive them from carbon dioxide of the atmosphere. So during photosynthesis by autotrophs, or either directly or indirectly. Heterotrophs, they use organic molecules that is formed during photosynthesis. So mean that it's very important. So photosynthesis will also help to purify our environment, as they are taking the carbon dioxide, and again they will be giving us oxygen. So it means they will help us to maintain a constant concentration of oxygen and carbon dioxide. Now there are different experiments to test if there is a starch in our leaf. So this is the aim, to test if there is starch. Materials we need a fresh green leaf, we need Bunsen burner, triple stand, test tube, we need petri dish, we need beakers, we need water, we need iodine solution, then a faucet, dropping pipette and alcohol. What are the methods? You pluck a fresh green leaf from the plants in the sunlight, make sure it's in the sunlight. You boil it, boil the leaf in water for 5 minutes so that it can kill the protoplasm and make sure the enzyme is inactivated. So then you and burst any starch grains that is present. So just place the leaf in a tube about 1/3 full with 70% alcohol. So place the tube in a water bath and warm the water gently until the alcohol begins to simmer. The alcohol will decolorize the leaf, so dip the leaf in warm water to soften it. Then place the leaf on a petri dish that has dilute iodine solution for 5 minutes. What will you observe? If the starch is present, you will see that in the leaf it will turn to blue-black. But if not, then there is no starch. So this shows that leaf has been photosynthesized. There's another experiment we have, an experiment that will demonstrate that oxygen is always being released by green plants during photosynthesis. To show that oxygen is released during photosynthesis, get 2 beakers, 1 liter beakers. Get a water plant, get a glowing splint, get water, get funnels, 2 funnels and 2 test tubes. So what will you do? That water plant, place it in a beaker that is containing fresh water and you cover with an inverted funnel and label it A. The same thing, you do it with a control experiment, label B. Only that while A is exposed to sunlight, B is covered with cardboard paper and leave them for some time. So let's see what will happen, the one that is exposed to sun and the one that is not exposed. What will be your observation? You begin to see bubble of gas in that experiment A, the one that is exposed to sun. On passing a glowing splint at the test tube end of A, you will see that it will be rekindled, showing that oxygen is there, making but no bubbles were noticed in B, meaning that there is no oxygen there. So, and it will not rekindle your glowing split. So the conclusion is that gas that is involved in A is oxygen, since it was able to rekindle the glowing splint. So this confirms that oxygen is produced during photosynthesis. There's another experiment to show that green plants require sunlight for photosynthesis. How will you know that? Yes, green plants need sunlight for photosynthesis. Method: take a potted plant that is well watered, discharge the leaves by keeping the plant in a dark cupboard at least for 24 hours overnight. Test the leaf sample of the plant for starch. The result is negative, yellowish brown color, which shows that there is absence of starch. So a leaf of this plant is partially covered with black paper on which a design pattern is cut, and the plant is then exposed to sunlight for 4 to 6 hours. So retest the leaves from both plants for starch. So what will be your observation? The one you cover, that covered leaf part, will show you brown coloration, while the one you expose, the exposed leaf part, will show you blue-black coloration. So the result, starch is present in the area of that leaf exposed to sunlight, but absent in the leaf part covered with black paper. So the conclusion is that the area of the leaf that is exposed to sunlight gave a positive iodine test, which indicates that sunlight is necessary for photosynthesis. There's another experiment that shows that water is needed for photosynthesis. To show that water is needed, get an alkaline pyrogalore solution, a conical flask, a Ritter stand, and split cork. Take a potted plant and discharge. That means try to take starch away from it. Discharge it by keeping it in the dark room for 24 hours. A leaf of the discharged potted plant, enclose it in a conical flask containing alkaline pyrogalore solution. This solution will absorb water. So the other leaves serve as control. So place the setup outside under bright sunlight for about 6 to 8 hours. Detach the experimental and control leaf from the plant and go and test for starch. Observation: the experimental leaf turns yellow. So the experimental leaf turns yellowish brown, negative test for starch. So while the control turns blue black, that's positive test for starch upon testing for starch. The conclusion is that this shows that water is needed for photosynthesis. So in our next class, we'll be looking at another topic which is under micronutrients and macronutrients. So thanks for listening.